And there. Okay, I didn't think I had any audio. All right, so the first one we're going to talk about is proteins. And we're going to talk about proteins first because they are the largest and most diverse macromolecule group. And it's going to take us the longest to talk about them. Proteins are responsible for the majority of the functions that go on in your body. Okay? They are enzyme catalysts. And what's a catalyst? What does a catalyst do? When did we talk about catalysts last time? They can help control a chemical reaction, and by control we mean speed it up or slow it down. If a catalyst is present, the reaction goes faster or slower? Faster. Good. So proteins can act as enzyme catalysts. They are used in defense. That means your immune system uses the presence or absence of proteins to identify foreign material. They also use proteins to help fight foreign material. Transport. Proteins help get oxygen and carbon dioxide in your blood to your tissues, to your systems. They transport waste and other nutrients, so they help with transport. Support. Proteins can be used to make things that support cells, that support tissues. Proteins are used to support muscles, bones, so they can be a structural component. Motion, of course, that's your muscles. Your muscles are mostly protein. The small filaments that make up your muscle are made of protein. Regulation, the endocrine system, which controls blood pressure, body temperature, blood sugar, hormones, these are sometimes proteins. Okay, so proteins help regulate your body. And finally, storage. Okay, when you store sugars, complex carbohydrates, fats, proteins are involved in that process, sometimes with the conversion of the material and sometimes just with help to make that supportive framework. This is a chart that lists the same things we just talked about, just a little bit more specifically. So again, you're at home, you're looking at this slide, and you can't remember specific examples that we just talked about. You can look at this chart because it's going to list them all again, and it's going to give you specific examples. Building proteins. This takes us back to building and breaking macromolecules. What is the process by which we build a macromolecule? It means making something by losing a water. Dehydration, synthesis. Dehydration synthesis is how we make a macromolecule, and there is no exception in proteins. We take the monomer of a protein. Mono means one. Okay, a single unit for proteins, that is an amino acid. Okay, amino acids can be called amino acids structurally because they contain two groups. What's called an amino group, which is a nitrogen and two hydrogens, and a carboxyl group, which is a carbon, two oxygens, and a hydrogen. If one chemical structure contains those two groups, we can call it an amino acid. Okay, so this is an amino acid, this is an amino acid. Okay, when we link two amino acids together, notice we're doing it through that hydroxyl and that hydrogen. We have a water that leaves. So it's dehydration synthesis that links these two these amino acids together to make a polypeptide chain. The bond that exists there is a covalent bond. Okay? So the covalent bond that links two amino acids together can also be called a peptide bond because that's what we're making. When amino acids come together, it begins to be called a peptide. Here is the protein hierarchy from the smallest unit to the largest unit. The smallest unit, the monomer, again, the single unit is an amino acid. When you start to link amino acids together, you get a peptide. When you put peptides together, you get a polypeptide because poly means many. Okay, many peptides together is a polypeptide. And the largest unit, of course, is made by linking polypeptides together, and those are proteins. Okay, so when you build a protein, that's what has to happen. When you break down a protein, when you eat chicken or steak or drink milk or eat cheese or any type of protein, even coming from vegetarian sources, you're starting with a protein and your digestive system has to chop it into polypeptides, then chop it into peptides, and then chop it into amino acids. Then you can bring it into your bloodstream and use it as a nutrient. So this goes both ways depending on whether you are building a protein or breaking it down. You have 20 amino acids from which you make every single protein in your body, which is pretty amazing because you have a lot of proteins in your body and only 20 building blocks. So every single protein in your body is the combination of these 20 amino acids. And I don't want you to remember their names, okay? I'm not going to quiz you on them, but you need to realize there are 20 amino acids. And most of them are given away because they end in INE. Alanine, leucine, isoleucine, phenylalanine, glutamine, okay, threonine. They all kind of end in INE. There are a few exceptions. Tryptophan, okay, that doesn't end in INE. No, you don't need to know the names. But I'm planting this seed because if you stay in anything, even biology related, you will come across these 20 amino acids again. So I just want to expose them to you. These are just special function ones, proline, methionine, and cysteine. So there are actually 17 on this chart, and here are the last three, 18, 19, 20. Okay. Protein structure. When we begin to link amino acids together and have polypeptides and have proteins, it's not linear. They don't just all go up in a line. They begin to twist and fold and go back on each other and link in different ways that gives the protein a structure of its own. Protein structure is incredibly important to the functioning of that protein. If it doesn't structure itself correctly, it will not work. And while that seems like, okay, the protein doesn't work, malformed proteins cause disease. So you can imagine that the study of protein structure is a huge field of study because it can even relate to some diseases. So we have different ways of categorizing protein structure. Primary, secondary, tertiary, or quaternary. That's a kind of fancy way of saying what? 1, 2, 3, 4. Okay? Primary, secondary, tertiary, and quaternary is just a fancy way of saying 1, 2, 3, 4. Let me grab a marker. Of course, yesterday I had a marker that worked. Okay, there's one. And the way that you indicate the difference between 1, 2, 3, 4 and primary, secondary, tertiary, quaternary is by putting a little degree symbol next to the number. 1 with a degree symbol means primary. 2 with a degree symbol means secondary. 3 with a degree symbol means tertiary. 4 with a degree symbol means quaternary. And this will not be the first time you see this or the last time, okay? The other type that we'll see how they fit in here when we talk about protein structure are motifs and domains. Motifs and domains fit in between primary, secondary, tertiary, quaternary, and as we start to go through, we'll see where those fit. First thing, primary structure. This is protein structure on the smallest level, which means what is the sequence of amino acids? If I start to give you something like alanine, leucine, tryptophan, methionine, if I start naming those enes, those amino acids, that means I am giving you the primary structure of a protein. Okay? It is the sequence of amino acids. That's the smallest level that we can evaluate that protein. If something goes wrong on primary structure, that means maybe it should have been leucine, tryptophan, methionine. But in our diseased protein, we found leucine, tryptophan, tryptophan. Okay? The primary structure was wrong. Here's an example of primary structure. Again, just generically, they are indicating amino acids. We see the carboxyl group and the amino group, and then the R is the functional group. That's what makes each amino acid unique. So the sequence of amino acids is the primary structure of a protein. Okay, secondary structure. When you start linking those amino acids together, hydrogen bonding starts to play a role. The different hydrogens begin to be attracted to each other, to other molecules, and that begins to change the shape of the amino acid sequence. Most notably, two shapes will arise, what's called an alpha helix or a beta sheet. An alpha helix is a helix, okay? It is like a corkscrew. It goes around and around and around. A beta sheet, okay, is a sheet of amino acids that looks like a little mountain range. Like if you drew a mountain range on a piece of paper, it's up, down, up, down, up, down. So if we look, here's secondary, here is a beta sheet, okay, again, it looks like this kind of corrugated look. And then here's an alpha helix on the right. It goes around and around and around. What else do you know that takes a helical shape? DNA. DNA also takes on a helical shape, and this is not unlike that. The helix is caused by the little hydrogens that stick off. Notice all the hydrogens in the primary structure. They are available for hydrogen bonding, so they begin to become attracted to each other, attracted to other molecules with partial charge, and they bend that protein into different shapes. Here's where we run into a motif. So motifs occur kind of between secondary and tertiary. So primary structure is what? The amino acid sequence. Secondary structure starts to take on two shapes. What are the two shapes? Alpha helix and beta sheets. A motif happens when you get patterns of secondary structures. Beta, alpha, beta. That means we see a beta pleated sheet, then we see an alpha helix, then we see another beta pleated sheet. A beta barrel is a bunch of beta sheets lining up in a circle. Alpha turn alpha, okay, or alpha turn means we have an alpha helix, and then the protein turns on itself and goes into something else, usually another alpha helix. So we're still talking about secondary structure, but now we're seeing repeating patterns of different secondary structures. Okay, so here's one beta, alpha, beta, beta pleated sheet, and then it turns into an alpha helix, and then back into a beta sheet. Here's alpha turn alpha, alpha helix, then the protein takes a turn and goes into another alpha helix. Okay, so patterns of secondary structure give you motifs. Tertiary structure is the final folded shape of a single protein. Okay, and that's going to become important as we get to the higher levels of protein structure. Tertiary shape is the final folded shape of a single protein, okay, or a globular protein. That means one protein by itself. The motifs, those patterns of secondary structure, and the nonpolar sections. What is nonpolar? Can it interact with water or not? It cannot. And what's the other word we have meaning not wanting to interact with water? Hydrophobic. The hydrophobic parts of the protein go towards the inside, which should make sense because these proteins exist in your body and you are mostly made of water. So if there's a molecule that's in mostly water, if it wants to get away from water, the only place it can go is deep inside itself. So the motifs and the nonpolar sections fold towards the interior of that protein because they are hydrophobic. And here is an example of tertiary structure. I see a lot of things. I see secondary structure, I see different motifs, but what I am viewing in this shaded square is the entire protein from beginning to end. I'm taking it all in, and that's the tertiary structure. It's looking at the entire protein, every part of it from beginning to end. That's the final folded shape. We still have two more to get to. Okay? Inside the tertiary structure, there are parts of the protein that can function independently. And those are called domains. So domains kind of happen in between tertiary structure and quaternary structure. Even though we have one big protein, and it's a single protein, that doesn't mean the whole protein does one job. Part of the protein can act independently and maybe interact with one cell, while another part of the protein can act on its own and interact with another cell. Okay? And these are called domains. When we find a huge single protein that has individually functioning units, we call those domains. Okay? And domains are based on function. Okay? They have to function independently, not just because they look independent, they function independently. And here are domains. So we're looking at a big protein. Here's the beginning. Okay? And then if we follow it, here's the end. But what we see when we look at this protein and the way it folds and the way it functions is that this part of the protein is independent of the blue part, and the blue part is independent of the pink part. And by the way, the pink part is a beta barrel. This is a bunch of beta sheets lining up in a circle. Okay, so I have beta, alpha, beta. I have alpha turn alpha. Okay, I have another alpha, beta, alpha. These are all different domains. Each one folds independently of the other and has usually an independent function. Last one is quaternary structure. Quaternary structure only applies if you have several proteins coming together to accomplish a job. Okay? So all proteins come together to form a functional unit, so a single protein actually acts as a subunit, a smaller part of a larger unit. Okay? An example is hemoglobin, the proteins in your blood that help you carry oxygen. You have to have 4 globin proteins to carry oxygen. If you have 3 globin proteins, you can't do it. 2, 1 won't carry any oxygen. You have to have 4 of them together. Hemoglobin has 4 subunits, 4 individual proteins that must come together. So when we look at quaternary structure, I am looking at the entire functioning unit. Okay? Everything shaded in blue is the functioning unit. But I notice that it's actually made up of 4 individual proteins. That's what quaternary structure is. So if I only ever have a single protein that does its own job, has its own function, will I ever see quaternary structure on a single protein? No. You have to have multiple proteins coming together in order to see quaternary structure. Okay. That's how we classify the structure. Now we're going to talk a little bit more about the folding. So proteins need, again, to be folded correctly in order to function correctly. Proteins fold according to hydrogen bonding, polar interactions, and what are polar interactions? What are they interacting with? Water. So how can we classify the polar part of the protein? Hydrophilic. The hydrophilic parts of the protein, non-polar interactions, which are hydrophobic, and also there will be something called chaperone proteins, smaller proteins coming around that large protein and forcing it to fold. All of these things have to come together perfectly and fold that protein absolutely correctly. If it is messed up, things like cystic fibrosis, Alzheimer's disease, these things can be have a function to the or have a component to the disease that includes the misfolding of a protein. Okay, so not only do you have a protein that doesn't work, but if you fold a protein incorrectly, it could actually cause a disease. Protein unfolding. Okay, so proteins do have to be unfolded sometimes when we're not going to use them anymore. And proteins can unfold by 2 means, what's called denaturation and dissociation. Denaturation is when a protein unfolds in response to a negative change in its environment. Okay? And when a protein has been denatured, it will never, quote-unquote, renature. Okay? It usually will never go back to folding itself correctly. It's done. Once a protein's been denatured, it's biologically inactive, it's useless. An example, an everyday example, almost everyone in here, unless you're a vegan, has denatured a protein. Okay? Eggs. Eggs are protein. Okay? The egg white is a protein called albumin. Albumin is actually one of the most abundant proteins on this earth. When you crack open a raw egg, what color is the egg white? It's clear. But when you put it on a hot frying pan, what happens to it? It turns white. The only reason it turns white is because you are denaturing it with heat. You are causing that protein to unfold, and when you change the structure of something, you change the way light hits it, and so now it's white. Okay? If I take that egg off the frying pan and I put it back in I shove it back in the shell and I put it back in the fridge, is it ever going to be clear again? No. Once it's denatured, that's it. It's never going to just refold itself magically back into a functioning protein. Okay, so when you denature something and our negative change would have been the heat of the frying pan, then you make it biologically inactive. This is also the same reason why you can't run a high temperature. You can't survive at temperatures of 105, well, you can do 105, 6, 7, 8, 9, about 110 you die. Okay? The reason is because every time your temperature gets higher, you're denaturing more and more and more of your protein. What do you think your brain is made out of? Protein. So when you start, then they say you're doing brain damage with a high fever. This is what you're doing. You're denaturing proteins in your body and you're ceasing your body's function. Dissociation only applies when you have several proteins functioning together, which means they have what kind of structure? They do have primary structure, but they have motifs, but they have... If I have several protein subunits coming together, then what kind of... Quaternary structure. Dissociation only applies when you have quaternary structure, several protein subunits coming together. When you start to pull those subunits away from each other, that's dissociation. You recycle your red blood cells at a rate of 3 million per second. Okay? So your body has a good recycling mechanism to pull that hemoglobin out, pull it apart, recycle it back into a new red blood cell. It disassociates your hemoglobin by pulling those protein subunits apart. Okay, proteins done. Now on to the next macromolecule group, nucleic acids. Okay? Nucleic acids can be summed up as your body's storage device. Okay? If you had a hard drive like a computer, it would be nucleic acids. Okay? This is your body's way of storing information. All instructions for the making of you, for the functioning of you, are stored in the nucleic acids. And you have two types. You have RNA, which is called ribonucleic acid, and DNA, which means deoxyribonucleic acid. The difference between those two molecules is in the name. Deoxy. D means if I decapitate you, what am I doing? I'm taking your head off. So D means to take away. Oxy, oxygen. Oxy means oxygen. So ribonucleic acid has something that deoxyribonucleic acid does not. What is it? It has an extra oxygen molecule. That's the difference between those two molecules. RNA, when you hear the word RNA, the next word that should jump right into your mind should be protein. Okay? RNA is only for making protein. The only time you would ever produce RNA, use RNA, make RNA, anything having to do with RNA, it has to be with a protein. DNA is for hereditary information. DNA is what you got from your parents. It's what you will pass on to your children. Okay? The DNA for making you is in every cell of your body. Okay? The monomer of a nucleic acid is called a nucleotide. What is the monomer of a protein? An amino acid. Very good. Amino acid, make sure you know your monomers and your polymers of every one of these groups. The monomer of a nucleic acid is a nucleotide. How do we know it's a nucleotide? It has these groups: a sugar, a phosphate, and a nitrogen. Over here is a picture of a nucleic acid. In blue, we have the sugar. And there's where we see the difference in RNA and DNA. Notice RNA has a hydroxyl. DNA just has a hydrogen. What's missing? The oxygen. That's why it's deoxyribonucleic acid. Here's the phosphate in yellow, and a nitrogenous base in orange, and that changes. Okay? We have a couple different nitrogenous bases, but we can say that each one has a sugar, a phosphate, and a nitrogen. Nucleic acids are chains of this 5-carbon sugar. So that means when I start to link nucleotides together, I do it through the sugar phosphates. Okay? We call it a sugar phosphate backbone. If you look at this picture, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and then these nitrogens stick out like fingers off of the sugar phosphate backbone. The nucleotides that make up DNA first, then we'll talk about RNA. DNA come in 2 categories, purines and pyrimidines. And the difference between purines and pyrimidines has to do with the different nitrogens that are on the group. Both purines and pyrimidines have sugars and phosphates, but they have different nitrogens. Adenine and guanine, which are abbreviated as A and G, and the pyrimidines, cytosine and thymine, abbreviated as C and T. Sequences of only these 4 nitrogenous bases, these nucleotides, make up your DNA. So whereas with protein, we had 20 building blocks to make every protein, your DNA, everything that is you and your functioning, is only made up of 4 different molecules: adenine, guanine, cytosine, and thymine. So if I were reading a part of your DNA sequence, it would sound like this: A, T, C, G, G, C, A, C, G, T, C, G, A. That's all I would see, is some repeating sequence of A's, T's, C's, and G's. Okay? You have 2 strands of DNA, and they make up 2 different helical shapes. So we call DNA a double helix. We saw single helices in protein. DNA always comes in a double helix. 2 strands of DNA always associate with each other and twine into a double helix. Here's a picture. Notice if we look at the backbone, we see sugars, phosphate, sugar, phosphate, sugar, phosphate. The nitrogenous bases from each strand stick out into the middle, and they associate with the nitrogenous base of the other strand. And that's what holds the 2 strands together. Okay? Those nitrogenous bases associating with each other. The 2 strands are said to be complementary to each other. One complements the other. It fits perfectly with the other because of the base pairing rule. And here's the base pairing rule. Adenine only bonds to thymine, and the opposite is true. Thymine only bonds to adenine. Cytosine only bonds to guanine, and the opposite is true. Guanine will only bond to cytosine. Okay? This is always true in DNA. So, now that you know the base pairing rule, I can give you a strand of DNA, one of the strands, and you should always be able to tell me what the sequence of the other strand is. Because if I have an adenine, it's always going to bond to a thymine, so I would see a T here. And if I have a thymine, it's always going to bond to a adenine, so this would be a G, G, C, A, T, C, C, G, A. Okay? You know that because of the base pairing rule. So if I give you a single strand of DNA, you should always be able to tell me what the opposite strand is. There's a typo in this. This last T should be an A. But otherwise it's correct. Okay, RNA is a little bit different. RNA is similar to DNA, but there are a few differences. First of all, we talked about one already. There's an extra oxygen. It has a ribose sugar instead of a deoxyribose sugar. There's no thymine. There are no T's in RNA. The T is now replaced with a U, which is called uracil. So the adenine from DNA will only bond with a uracil in RNA. RNA does not form a helix the way DNA does. RNA is linear. And what does linear mean? Straight line, okay? A straight line is linear. So RNA is linear, and RNA will always be single-stranded. You will never see RNA coming in a double strand the way we see DNA. Okay? How do we get RNA? RNA is only present for a short time in your cells. And the reason it would ever be present is because you need to make a protein. So your cell has to go what's called transcription and translation. DNA is always present in every single cell in your body, except for a red blood cell. DNA holds the key, has the blueprints for making protein. So if I want to make a protein, I have to go to your DNA, I have to open it up, and from your DNA I have to make a piece of RNA. That's called transcribing. Okay? When the cellular machinery undoes the two strands of DNA and reads one of them for protein instructions, the product of that is a strand of RNA. Okay? Then that RNA will be taken by some other machinery. It will be read, and we'll see AUCGGUACUA, and that will be translated into a protein. Then we get rid of the RNA. We trash it. It goes right in the trash can. We degrade it. RNA is transient. It's here as long as we need it, and then we get rid of it. DNA is always present in your cell. The reason DNA going to RNA is called transcribing is because transcribing is in the same language. If in class your hand is flying, you're taking sloppy notes, and you go home tonight and you rewrite your notes neater so you can read them, that's transcribing. Okay? You're repeating the same language. DNA and RNA are the same language. RNA going to protein is translation, because if I need somebody to come in and speak Chinese for me, what do I need? What is that person? It's a translator. Translators do different languages. RNA and protein are two different languages. One's amino acids, the other one is nucleotides. So that's why DNA going to RNA is transcribing, RNA into protein is translating. Again, RNA will only be present when you need it to make proteins. So, if I give you a strand of DNA and I say I want the corresponding strand of RNA, you should also be able to give it to me. So across from this A we should see a U, because there are no thymines in RNA. And then from this T we should see an A, G, C, A, U, C, C, A. Okay, and again, typo. Pardon me, that should be an A. Okay. That is the strand of RNA that corresponds to the strand of DNA I gave you. Now, if I were the cellular machinery wanting to now make a protein based on this strand of RNA, I no longer read it one base pair at a time. Now I take 3 at a time in what's called a codon. If I want to figure out what part of that RNA corresponds to an amino acid, I have to take 3 at a time. UAG corresponds to methionine or threonine or tryptophan. Then GCA, then UCC, then G, and that should be an A, something else. I am reading 3 at a time to figure out what amino acid I need. And here's a chart of every possible combination of the 4 bases in RNA and how you can put them into codons. You don't need to know this chart. You don't need to memorize it, nothing. Okay, but I just want to show you how we know this. UUU, phenylalanine. UUC, also phenylalanine. You'll notice very quickly that the 20 amino acids each have several codes. Okay? Glutamine has 2 codes. Asparagine has 2 codes. Lysine, 2 codes. Some of them have 3 or 4 codes. Threonine has 4 codes. Alanine has 4 codes. So every time the machinery runs into GCU or GCC or GCA or GCG, it knows it needs to make an alanine. Okay? And it plucks an alanine and a threonine and a tryptophan and a spargenine, and then it's linking together amino acids to make a protein. The one thing I do want to point out is starts and stops. There is a specific codon that means start making the protein here and I'm done. Stop making the protein. Here are your stop codons. UGA, UAA, UAG. These mean stop. The start codon is AUG. That is also the only codon for methionine. So what can you say about the first amino acid of every single protein in your body? Every single protein in your body has to start with methionine. It all starts with AUG because there's only one start codon and there's only one methionine codon, and they are the same. So no matter what the protein is, whether it's a piece of hair or a cellular structure, it doesn't matter what it is. They will all start with methionine. Okay. ATP is a molecule that we'll talk a lot about in cells, and you'll run into it again. It stands for adenosine triphosphate. Tri means 3, so it's an adenosine in 3 phosphate groups. ATP is the main source of cellular energy. Okay? This is similar to the way that our society functions off money. Okay, you need money to make transactions and get goods and make purchases. Your cell needs ATP to do anything. Okay, if it wants to do a process, a function, take something in, get rid of something, every time it needs ATP. Okay, adenosine triphosphate. Adenosine looks an awful lot like another word we were talking about. There it is, adenine. Adenine and adenosine are very close. So the group of macromolecules that ATP falls under are actually nucleic acids. So if I ask you on the test, and I will, what macromolecule group does ATP fall into? The answer will be nucleic acid because of the presence of adenosine. All right, 2 more groups, and then we're done. Lipids, okay? Lipids, fats, oils, that's what we're talking about in this group. The main characteristic of a lipid, it's insoluble in water. What other 2 words do you have that describe that? Hydrophobic or nonpolar? Nonpolar. Hydrophobic, nonpolar, insoluble in water, these all mean the same thing. Do not want to have anything to do with water, can't interact, can't form hydrogen bonds. That's a characteristic of lipids. One important group of lipids that's going to haunt you when we start cell physiology are called phospholipids. Okay? They are made up of a glycerol, fatty acid chains, and a phosphate group. Okay? You will see them as this ball and stick model when I start drawing them. But they have a phosphate and a glycerol head, and then we see 2 long chains coming down. That's a phospholipid. It makes up every cell in your body, phospholipids. Okay, another important group of lipids, fats. Fat molecules are called triglycerides. Tri means 3, and we're actually just talking about 3 fatty acid chains, not 3 glycerols. Okay, 1 glycerol head, but 3 fatty acid chain tails. Instead of a phospholipid, it has 2 fatty acid chain tails. Again, don't forget that it doesn't matter what the name of the group is, carbon's always there. Okay, carbon has been in proteins, it's been in nucleic acids, and it's here in fats again. Carbon molecules determine the saturation of a lipid. A saturated versus an unsaturated fat. Saturated fats have the maximum number of hydrogen atoms attached to every carbon. Unsaturated fats have double bonds occurring between 1 or more pairs of successive carbon atoms. Let me just show you a picture of what this means. Here's a saturated fat. Notice every carbon will be linked to a carbon to its right and to its left, which still leaves 2 bonds. So those 2 bonds are taken up by a hydrogen. The fatty acid chain tails appear very uniform and very straight. An unsaturated fat, if we look up here, instead of bonding with a hydrogen, we'll double bond with the carbon beside it. That puts a little kink in the tail because double bonds are tighter. So it puts these kinks and makes these fatty acid chain tails look very strange. This is an unsaturated fat because saturated means maximum, the maximum number of hydrogens. When it's unsaturated, it doesn't have the maximum number of hydrogens. It starts to double bond with itself. Other lipids, terpenes are a lipid that we use to make rubber. Steroids, estrogen, and testosterone, progesterone, the sex hormones, all come from cholesterol. If you want to make sex hormones, you have to have cholesterol. That's why there's good cholesterol and there's bad cholesterol, and also why fat has to be a part of a balanced diet. Here is a steroid. Okay, this is cholesterol. Your body would take this, it would chop off a couple things, and it would come up with the steroid hormone. Another one, prostaglandins, are a type of lipid. Prostaglandins are most notable for headaches. Okay, when you get a headache, sometimes it's due to a prostaglandin, and prostaglandins also are a lipid. Last group, try and go pretty quickly through carbohydrates. Carbohydrates can be defined as a molecule that contains carbon, hydrogen, oxygen in a ratio 1 to 2 to 1. So if I give you this formula, okay, carbon, hydrogen, oxygen, 6, 12, 6. Is that a carbohydrate? Yes. 1 to 2 to 1. Okay, what about this one? Carbon, hydrogen, oxygen, 4, 5, 4. No. What would this number have to be? 8. Good. Okay. So if I give you a couple carbon, hydrogen, oxygen molecules on the test, and I show you what numbers are present, you should be able to tell me, yes, it's a carbohydrate, no, it's not a carbohydrate. The simplest carbohydrate, the monomer of a carbohydrate, is a monosaccharide. Saccharide means sugar. So the mer, the unit of a carbohydrate, is a saccharide. When we put 2 together, we get a disaccharide. If we put a lot together, we get a polysaccharide. You store carbohydrates. Again, carbon is your body's favorite energy source, and when you give it too many carbohydrates, it does not want to let go of them. It wants to store them for later. So it takes a simple sugar and it turns it into an insoluble sugar. So if it's an insoluble sugar, what is it? What other words can we use to describe it? It all of a sudden becomes hydrophobic. It doesn't interact with water anymore. In plants, when plants do it, you get starches. What's a starchy plant? When you eat a starch, a potato or rice, those are starches that a plant has made. In animals, we call it glycogen. Okay, the glycogen in your body is an insoluble carbohydrate. When your blood sugar becomes low in between meals, you break glycogen into glucose. Other carbohydrates, cellulose, which you cannot break down, is in plants. Okay, cellulose can only be broken down by ruminants. Give me an example of a ruminant. What's an animal that can break down plant material? A cow, a horse, a sheep, a goat, a deer. These animals have a digestive system called rumination. They ruminate. They can break down cellulose. And then one of the strongest forms of carbohydrates is called chitin. Chitin is what you find a lobster shell or a crab shell or a beetle to be made out of. Very few organisms on this planet can actually digest that carbohydrate and extract any energy from it. All right, that's the end. See you guys at 11 for your histology quiz.